So I think it's a bit difficult task to close such a nice workshop. And also we have to wrap up everything, and we have everything in this session. So I prepared the session with Jozica, Gricar, and Peter Prislan. And I also use material from Gonzalo Perez de Lis and Ignatius Adicornia. The presentation is based on question, but we try also to make a general approach and to place the question where appropriate. So wood formation monitoring and xylogenesis was quite intensively used these last years because, again, to try to better understand the relationship between climate and tree growth, and also to better understand the relationship between wood anatomy and tree function. The limits are that at this time, gymnosperms were a lot more studied than angiosperms. And also the aspect about phenology, they were a lot more studied than other aspects we will also describe in this presentation. Okay, so in the presentation, we try to go from the sampling for wood formation monitoring to the data processing and data analysis, and also we will have a large part on the results and interpretation. And all along, we try to answer the question that were raised during the inscription. So we will start with basic information on wood formation, and also I place all along some kind of pause if you want to ask question. So you can again ask question in the chat, and I will stop at the end of each let's say chapter. So the chairman can report the question to us. Okay. Okay, this is just for very beginner. That just to mention that what I find nice is that the cambium, which is the living tissue which is forming xylem on one side and phloem on the bark side, I see it like a living envelope all around a tree. So usually we are studying a stem, and this is the main part of this talk. But don't forget that cambium is also present in roots and also in branches. And the functioning of the cambium is also related with the functioning of the trees, of course, with for example, it needs sugar to produce xylem and phloem. So you can see that this is the stem. This is the last ring with early wood, late wood, the cambium and the phloem and the bark. So all of this we saw it already. And then you can split again this the cambial zone with the young and old phloem and young and old xylem of the year, and then previous 3 rings. Okay, so I just go quickly on this basic introduction, but then if you again, you can ask questions and we can discuss that on the forum also. So now this is really the core of the process of xylogenesis. So we start from one cell of cambium, for example, a mother cell. This cell will divide it to create 2 new cells, and one of these cells will enlarge. Then the cell wall will be built. This is this phase, we call it wall thickening usually. And then the last phase of development of differentiation is the death, which is a programmed cell death of this wood cell. And then the wood cell is left with an empty lumen and its cell wall, and it gets its full property for functioning. Okay, this I talk already about the process, but yeah, don't just mention quickly that in the division, you already have the process of cell growth, primary cell wall formation, of course, and cell division. In enlargement, the process is only cell growth. In the phase we call cell wall thickening, there is secondary cell wall deposition, but there is also cell wall lignification. And right now, we are not really accurate on how to distinguish these two processes in this one, for now, one step. And then the last process, again, is programmed cell death. And what I found also very interesting is that this process, they are related to final cell properties, cell dimension, in a very strict way. And for example, the final cell size and shape is only related to cell enlargement. And the final amount of cell wall and most of its composition is only related to cell wall deposition. Okay, and this process then they, of course, they give the final function, like we saw them already, like conduction, support, and so on. Yes, I have to say that, of course, this is more trachea centered view, yes, but these differentiation phases, they are also true for all the xylem elements, but not with the same importance. Okay, and then we move in the more practical way. So I will go quite quickly because now I will review all we saw already in the previous section. But I will just insist a little bit on the differences. And you can see also it's nice because, of course, the same question appear again. Like, for example, how many trees should we monitor? And I would say that in wood formation monitoring, the problem is even stronger than in quantitative wood anatomy in general, because rather than one cut for one tree, we will make 30 or 40 sections per tree and per year. So it's even more problematic. So usually the number of trees is quite low, could go to 3 to 5 trees. And as Georg mentioned at the beginning of the presentation, this number is, of course, a bit too low, but it depends on our resources. But on the other way, I made some tests with 15 or 30 trees, and I end up with some evidence that 7 trees is already quite good to have a seasonal signal. Then, of course, it depends of what you are interested in. And then we have also new question like, shall we change our tree from one week to the next one, for example, or is it better not to do it? And also we have this question that all of us have, that usually we focus on the stem, but root and branches development is also very interesting. And then about environmental variable, again, it's a bit more difficult again, because now if we are really interested in the detailed development of the xylem, of course, we need more accurate variable from the site point of view. So we need, for example, the temperature of the stem probably, of the soil maybe, and also from the time step resolution, because we usually have weekly measurements, so for sure we need at least weekly measurements also for environmental variable, climatic variables, let's say. Okay, so this I I see that I had more questions than answers, but this is also to make you think, and this is also really connected with what was said several times before, that this really, everything depends on your question. So then the sampling of wood formation, usually there is several tools, but now one of them quite imposed all over the world, which is the Trefor tool, because it's quite easy to use and you can extract nice, what we call micro cores, so small cores. And most of the studies do that every week or bi-weekly sometimes, because this also depends a bit of the length of the growing season. Yeah, and also a problem we have in addition to the one we spoke about before is also the variability around the stem. Well, we mentioned already reaction wood, but here we have more than that. And because we need to compare all these small cores all around the year, the variability around the stem is really a problem for us. And then another problem which annoys us and which is new compared to quantitative wood anatomy or dendroecology is the reaction of the cambium. Because we are punching quite regularly, if the cambium is reacting quite strongly, in fact, we will start to measure the reaction we are creating. And for example, this is very visible on larch, like here, where larch is quite prone to create resin ducts. And so you can see that if you punch quite regularly and closely, you will register more and more resin ducts when the growing season is coming. So this is also something which is important, and we have to pay attention that we are creating a reaction, and we have to pay attention not to work on this reaction, at least, or on purpose. Okay, so this slide, I will skip it because we talk quite already long time on the sample preparation. But just to mention that this movies was prepared by Peter and colleagues, and you have the information on the QNet, I think, web page. Okay, and so this is just to summarize the approach, and we can already answer some question if you have some on this first part on sampling and methods. So we took microcores from the stem most of the time, but there is few studies on branches and on roots. You will have an example afterwards. Then usually we use the method is paraffin and embedding and rotary microtomes sectioning, but there is also other trial. And then microscope observation, light microscope observation after staining, but also here there is some new methods emerging. And then the idea is to reconstruct the growth all along the year, as you can see here. So this is a summary with 5 anatomical sections, but most of the time we have between 20 and 40 such anatomical section. Okay, is there any question from the chat? And then maybe I have also to mention that I didn't pay attention to the time. So you can make sign if at the end the talk is really too long.
Cyrille, there is a very quick question. They are asking if you remove the bark with the species with very hard and thick bark. You can answer very easily.
Yeah. This I can answer a quick answer. Is that in fact it depends a bit of the species, and I have in mind, for example, oak gets a very large bark, And often it's not possible to get a good microcores if you don't remove a bit of the bark. So with some species like oak, Scotch pine also, for example, I would say it's compulsory. But then you have to pay attention to that and not to do it too much. Of course, too much because you can harm the cambium, and then you have to change tree. And also not too strong and too long in advance, because my expertise is that you also created a reaction from the tree, even if you just remove the bark. So I would say if it is possible not to do it, for example, I guess it's possible on beech, then I would suggest not to do it. But on most of the species, we are obliged to do it. And then we have to keep it secure.
Okay, thank you, Cyrille. Please consider that you have a bit less than 10 minutes more to end up with this part.
Oh, this part. Okay.
With the last step.
Okay, I try to move. Okay, so this is the criteria, the anatomical criteria to make the classification of the different cell in the different phases. So, for example, this is cambial cells, this is enlarging cells, thickening cells, and mature cells. So the criteria, they are shown here, but I don't go into the detail. And maybe I give the floor to Peter, so you can, so you can present the equivalent on angiosperm.
Sorry, do you hear me? Thank you.
Yeah, fine.
Yeah, the difference, there is a slight difference between angiosperms and gymnosperms because there are many, because we have two different cell types. These are vessels and fibers. And we know that vessels differentiate slightly differently or have a different differentiation dynamics than the fibers. So similarly, as in case of conifers, we identify at the beginning, we can see cell enlargement, then we follow secondary wall formation. We could also detect the beginning of lignification. Usually lignification first starts in vessels and the surrounding cells. And this can be quite noticeable, especially at the beginning of the growing season. According to our experiences and studies, vessels can lignify 1 month earlier than fibers. And at the end of the growing season, we can also identify, for example, cessation of cell expansion and then cessation of xylem differentiation. This means that all cells at the end of the growing season are fully lignified and are able to perform their main functions. So back to you, Cyrille.
Yeah, I think it's Jozica now.
Yes.
Presenting the phloem differentiation.
Yes. Hello, from my side. I would just like to quickly mention that, as already Cyrille said, so cambium is bifacial meristem, and we are mainly talking about the xylem usually. But of course, because for us it is in terms of dendroclimatology, dendrochronology more important. But of course, for trees, it's not the same situation. Phloem part is extremely important. Also, if we want to study tree response to different environmental conditions or even stress events, I think that it gives very important information. And of course, it actually connects the carbon source, like leaves, to the carbon sinks, which is cambium. So in my opinion, information on the phloem dynamics, it gives you additional information of tree functioning. The phloem formation dynamics is a bit different. So I show here only basic one example on spruce from temperate sites. And well, it starts a bit differently. So the onset is with the development of initial early flowering sieve cells. So they start to differentiate without former cell divisions. Actually, they are formed in the previous growing season, and they start to grow maybe a few weeks before or just at the onset of cambial activity. And then what we follow is cell division, cell expansion, but not so much about wall formation because it's not so easy. It occurs, of course, but maybe light microscopy is not optimal. And for example, in angiosperms, wall formation, I think it's a bit even more complicated than in the case of conifers. Below on the bottom, you can see major steps. I think, Cyrille, there is one image more. So the first step is this onset of differentiation of first cell. And then we have on the fourth image transition from early phloem to late phloem. And you can see also that it is very easy in temperate sites to show what is the increment. And I will show you later that this is a bit more complicated if we go in different environments. So that's it for now. How wall thickening phase, how important it is. Yes, we use it. So for wall thickening phase, we use polarized light because I think that the staining is not so, in my opinion, is not so optimal. So whenever, also for the conifers in the phloem, you can use polarized light to detect it. Okay, Cyrille.
Okay, then this is just a presentation of the kind of raw data we can have. So this is for 3 species, pine, fir, and spruce. And you have here the day of the year and 3 different years and the different differentiation step, as we saw, cambial cells, enlarging cells, thickening cells, and mature cells. And then with this information, we can, of course, try to understand the relationship with tree structure and with environmental data. Okay, so this is mainly for gymnosperm, and it is also possible to work, for example, with angiosperm, with not the number of cells, but the bandwidth. But you have to take into account which type of cells you are considering. So it's a bit more difficult, but it's feasible. Then I developed a package for R, which is called Caviar, to help to process raw data. The package was developed for conifers, but it could work also for angiosperms with certain conditions. And then it helped to process the data and prepare plots. And yeah, and then we will see the different results. Also, I mentioned, because this was a discussion during our previous workshop, the development of a global xylo initiative, which is a collection of monitoring wood formation monitoring data. So for now, it contains only conifer trees, but we are trying to develop it now for angiosperms also. It's a different database for now, but then we'll talk about this initiative project and the development. Okay. So, and now, yes, this is just because today, I don't know if you are aware about that, but today cinemas, they are reopening in France. So it's very important for us. So you can see a short movie from Ignatius here. And in the meantime, if you have questions on this observation of xylem and phloem, you can post them in the chat. And then what I will do, because we are really late, I will just take one example per category in the following slides. Yeah, and we will keep the whole presentation for later discussion. Okay, so this was the reopening of the cinemas in France. Is there any question from the chat, or shall we move on?
Okay, yes, there are a few questions. One is about very quick about the use of the blue staining. I suppose it was Astra blue. And another one is if it is mandatory to use polarized light microscope. And there was another one. It was about the angiosperm. And there's a specific question about the eucalyptus, if it is because of the cambium zone tears soon after the collection. So how to avoid this? Okay.
So for the two first questions, I can answer very quickly that for staining now we use, there were several different methods at the beginning, but now I think we agree to use FastGam, most of us. So there is safranin and Astra blue in the same bath. And then this avoid a lot of losing section, difficulties and so on. So now this is quite standard. And then I forgot the second question.
The second question was about polarized light.
Yeah, polarized light is an important step. So it's not compulsory, but I would say for me, it's one of the most important observation, the one we are really sure about. So I cannot say it's compulsory, but it's very, very good help.
Yeah, and, maybe I can add a little bit, because polarized light helps us, especially at the beginning of the growing season, to separate the enlarging cells, which have really thin cell walls, from the cells that already have secondary cell wall, but it's still undetectable with the naked eye or with the microscope. So if you turn the polarized mode on, you can clearly distinguish this, and it's a really big help.
And it can be sometimes delayed, because if, usually red, it stains lignin, and you don't see because the wall thickening starts much earlier. It's still, it's carbohydrate, so it can be delayed.
We can also show the photo as Sabina suggested. Maybe you can go back to the slide I presented. We can clearly show how the differences between polarized light and conventional transmission mode.
Yeah, because we are really short in time.
Yeah, that's true. That's true. I totally agree.
But maybe this is a good thing to keep on through the QNet forum. We can go deeper in this. And the last question, very quick one, was about very quick forming hardwoods, how to avoid the tears of the tissue while collecting and before processing.
Yeah, this is very difficult to answer because it really depends on the whole process. First suggestion, it was made already, but the sharpness of the tools, it's really crucial in our case. So this could be something. Also depend the difference between the bark stiffness and the wood is also very important, and maybe can create some tears. And then not to go too quickly in strong baths of alcohol, because this also can tear the tissue apart. But the problem is, you see, you can have this kind of problem at every step of the process. So it's difficult to answer from, yeah.
If I can add something, sorry. Jozica.
Yeah, just I wanted to add that, for example, in ring pro species like oak in spring, we also have a lot of problems because they grow quite fast. You have these big vessels, they are not lignified, and yeah, it can be quite a challenge also for us.
If I can just add something, I have no experience with the eucalyptus, but with the Quercus ilex. And I think that it also depends on the period of the year in which you collect the sample. Sometimes it's mostly impossible to collect a very good sample, but you have to try. It depends on your species. Sometimes you just take a couple of micropores, the first fails and the second is okay. And then another thing is the fixation. Sometimes it is enough with alcohol, 70%. Some other times we found that a post fixation with other fixative worked better, but this depends also on the species. So the advice is to try which is the best method for your species.
I just wanted to add something more, because I know that eucalyptus has also a lot of reaction wood, and also maybe the fibers are under angles. So it's in these cases, it's even more problematic, because I remember that there were really some certain trees we had the problems. And later we saw that the tension wood, the fibers were a bit deviated and so on, so on. So I can imagine then you have problems.
Okay, and I think this is very interesting. We could stay here for long, but we will go ahead in the forum with these hints, and maybe I ask Cyrille and Peter and Jozica just to summarize and sum up with this part.
Yes. So what I will do, I will just present what we can do with this data. And I will just mention that concerning the first part, yeah, we can continue to have this kind of discussion in the QNet workshop. And yeah, we are also thinking to prepare more guides to help you. This was mentioned in the things to come. So I will just go for what we can do with this kind of data. So one possibility, which is the most which is the most used one, is to work on phenology. So just the circle just show that sometimes the cambium is dormant and sometimes the cambium, of course, is active. And then with phenology, right now we use critical dates, for example, to present different states of phenology in the wood formation in the cambium. Okay, so this is one big part of what we can do with this data. And by the way, I just highlight that we are working a lot on activity, but there is not so much on dormancy, and you can see that there is things happen even during winter. But this is difficult with usual microscope. Okay, and then I go directly. So sorry, Jozica and Peter, then I squeeze their slides. But maybe I can take this one to show about the dynamics. So this is the second thing we can do with this kind of data, is to show the dynamics. And I choose this slide because you can see the dynamics of the xylem, so the accumulation of the number of cells, and also the one in phloem. And then you can also see that this was for cold areas, for example, where you have this kind of bell-shaped curve for the rate of growth. But in Mediterranean area or different area, you can have a bimodal, for example, curve for the rates, or at least different things. So these dynamics will depend of course, the species strategy, but also the environment. And we can have it for xylem, but also for phloem. Jozica or Peter, do you want to have a word on this?
Yes, and just a short word, because I think this is very important for people who are not coming from temperate or cold climate zones, because not here that how, of course, the dynamic of the xylem is important, but here what is also important is that the timing of the xylem and phloem formation does not start at the same time as we are used to at the temperate side. So the division starts on the phloem xylem at the same time, but in Mediterranean area, we don't know what happens in the phloem. Obviously, they grow all the time. So the dynamics is totally different. The structure is confused, so you don't have 3 rings there. You don't see the boundary. So the rhythm of the cambium is totally different because they don't have true dormancy. And I think this is really important for people coming from different areas. It is more complex, and it really shows you how the radial growth of the cambium rhythm can really adapt to certain environments. So it's not so simple, but it gives you an idea how plastic can it be. So I think this is very important part. And you see that it's red and orange in the flowing, because it is just imagination. We don't know if it's true, you know, so it's just what we think it happens. And also one point, that even though the cambium is dormant, cell differentiation can still continue, and it does in wintertime in Mediterranean. So you don't have annual increments when you do this dendroclimatology. The data that you use for the climate, they can be different sets.
Okay, thank you for this. This is very important, Jozica. So it's time from wrap up, but I would just to ask you, Peter and Cyrille, and maybe also myself, the time is ripe for having a project, a QNet project on this issue. So we'll keep going on this with your help, hopefully.
Yeah, definitely.
And then last point is about what I call the kinetics. So with this data, we can also compute the cell residence time in the different phases and the rate of differentiation. This is a schematic example here, where you have the time the cell took in blue, the time the cell took to enlarge, and in red, the time it takes to develop its cell wall. And then with this information, we can try to understand the trimming structure. For example, we already spoke about wood density. So how wood density is related to this wood formation kinetics. So this is, for example, an example for gymnosperms. And for angiosperms, even in case of beach where it's quite difficult because the structure is homogeneous, we can still try to understand the difference of vessel size from the beginning of the ring to the end related to these parameters. And finally, I will just move for one last slide about the relationship between the growth in the different parts of the trees and also between leaves, roots, and stem, because this is also a huge avenue of research. And there is work on this, but not so much, and we need it more, even if it is very time consuming.
Yeah, because if I just add something, that usually we are talking about cambial activity and try to link it with leaf, we always talk about stem base, but it's not the same in different tree parts. So this information, I think it's crucial, because in this case, in Quercus pubescens, we show that in branches is different dynamic, narrower rings, and you can see, and 1 month, approximately 1 month shorter duration of cambial activity. So different tree parts can reveal different information, I would say.
Okay, thank you very much. So I think now Georg can just wrap up everything, because we already had the question during this session. This was very, very nice to have this interaction with the continuous question. So thank you very much to the three of you.
Yes, thank you also from my side. And this brings us really to the end. So a few last words to close this workshop.